Hello, everyone. Thank you for joining our webinar today titled Supercritical Fluid Extraction and Chromatography, the Critical Promise for a Greener Tomorrow. I'm excited to have Richard Sanders and Marisa Feller here with me today hosting the webinar. I am Amy Knock, and I'll be moderating the session. Before we begin, I'll let you know more about how this webinar will run. The webinar is being recorded and the slides and recording will be available for you within 3 business days. A short Q&A session will follow the presentation to answer any viewer submitted questions. During the webinar, you can submit your questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. Remember, you can submit questions all throughout the webinar. That concludes my brief introduction today. I will now pass the webinar over to you, Richard. You may begin your presentation.
Thank you, Amy, and thank you everyone for attending today. We do appreciate your time. This is not going to be a terribly long webinar. We are going to be talking about some fairly historically old technology, which many of you will be familiar with, but what we would like to highlight is some of the new updates and how we think this is really the way the industry is going to be heading soon. So we'll start by letting Marisa introduce herself, and then I'll introduce myself.
Hello and welcome. My name is Marisa Feller, and I am the Vitamins Business Unit Manager for the Madison, Wisconsin Eurofins site. And I'm very excited for Richard to give a brief summary and introduction to this technology and how we will be utilizing it within the vitamins lab in Madison.
And I'm a business development director for Eurofins, and I actually represent all of the Eurofins U.S. food laboratory network, so all of our chemistry and microbiology units, as well as having access to our global network of laboratories. So our conflict of interest transparency statement, we're not endorsing any specific instrument company over another today. We will be touching on a few. We're not representing any government or industry or academic group other than the Eurofins Laboratory Network. And we have a bias. We love food and drink, and we take supplements, and we care about the planet as much as or more than the next person. So this testing for life is a real thing to us. So for those that are not familiar, and those that are, please bear with me, who is Eurofins Scientific? So Eurofins Scientific was founded back in 1987. And I jokingly say that because that's also the year I graduated from high school, around the SNF's NMR proprietary technology. And this technology was actually developed to detect adulterants in the wine industry, which was a really big thing in the late 80s. And the companies continued to develop that technology, offering a wider range of products around that original platform, and then continue to grow over these last 30 plus years, building on its roots of innovation and research to the global network of laboratories that we are today. And just to give a little size and scope of the Eurofins group, currently we have just over 900 laboratories in 50 countries with around 55,000 members of our team, with over 200,000 analytical methods, all tested in just around 10.8 million square feet of laboratory space. And each year we complete around 450 million different tests, whether those are around safety, identity, composition, authenticity, mineral tracing, purity of biological substances, environmental and microbiology controls, and other clinical diagnostic services. And the presentation today is going to nest very well right in the middle of our critical vision, mission, value, and sustainability promise. You can read a little bit more about this on eurofins.com/about-us. But we really are committed to to contributing to a safer and healthier world by providing these new technologies, providing the customer service on how you can take advantage of these technologies. And when we're viewed as a leader in the industry, that means sometimes we need to do things that are important as far as change goes because it's the right thing to do. And we believe that this technology really has achieved the right time, the right engineering, and now is really the right thing to start looking at as a viable and actually better alternative to existing chromatographic methodology. So quick overview on the objectives of what we're looking to do today is explain why this is such a fast technology. We'll give you just a real small history on supercritical fluid and some introduction, give you a name to remember that should make you smile after this, show you why we believe this is a better technology from a perspective of being the ultimate green chemistry, and why the method is really a better way to go, in some poster sessions that we've already given at some conferences that we've been attending. And it is uncompromising technology. So we're not asking you to take any hits for the team by looking into this or by changing anything to take a step backward. Everything today you're going to see is a step forward, whether it's in accreditations, how we compare samples between existing historical and what we would do with this technology. You'll see that we are cutting zero corners and offering you only upside with this technology. So to start out with, it's a little quiz, I guess a little game, if you will. Keep in mind as we go through the slides, what do these three images all have in common? And after this first section, I'll come back to this slide and tell you where all these dots connect. So a little brief history on supercritical fluid. For those of you who have a degree in chemistry, you've seen this already. For those of you who've taken high school chemistry or any other of the sciences that required you to take 1 or 2 semesters of chemistry, I am going to explain nothing new because you should have seen it by week 3. Supercritical fluid is a state that's achieved by taking any substance at a temperature and pressure above its critical point, which you see there in red. So this is a phase diagram with pressure manipulation, temperature manipulation, and you can see how a compound can, at varying degrees of those manipulations, exist as a solid, as a liquid, or a gas. The triple point is at that phase where they all exist in equilibrium, and as you continue to increase pressure and temperature, you can manipulate it to a point which is called a critical point, and at that point, the fluid and gas phase then go into a supercritical state where it starts to have characteristics of both gases and liquids simultaneously. This chart to the right will show you rates of diffusion, density, and viscosity, and why these are important, or these are the parameters that we manipulate as we're starting to fine-tune chromatography, choose which chromatographic system is best for us, and these are the parameters that we look at as we're developing those technologies or trying to use that. So the key here is, in a supercritical state, fluids can diffuse like solids, they can diffuse through solids like a gas, but then dissolve materials like a liquid, which is really an important point. Close to that critical point, however, any variations in pressure and temperature results in manipulations of the chromatography down the line, or what can be used as fine-tuning. Also, if it's off, things will quickly go south. So what does this all mean? Is in a critical state, we can really take advantage of faster flow rates and lower pressure, right? So it dissolves better, it moves faster without pressure concerns as we start to manipulate things that we traditionally seen in other forms of chromatography. So we can literally take hours and turn them into minutes. There's actually several compounds that can be used in supercritical fluid technology. And here it's just a small example of some. And you see that I have CO2 or carbon dioxide highlighted. You'll notice that it's literally the perfect choice. The temperatures that need to be achieved, 31 degrees C, and the pressures that need to be achieved are both relatively low, right? So we're talking around 80, 87, 85 degrees Fahrenheit, and then again, relatively low pressure. And CO2, of course, is non-toxic, it's non-flammable, and we all know controlling this particular gas, right? It's in an abundant supply, but it's also easily removed from the analytical environment. It's also has GRAS status, right? So it's generally considered to be safe. It's used in food and pharmaceutical applications. But you also see nitrous oxide has very similar characteristics, but it does not have that same non-toxic effect, right? So it's also slightly more rare. So anytime you start talking about safety issues, like everybody breathing in laughing gas and then having something rare, there's cost and safety associated with it, really. So there is no real second place to carbon dioxide, and that's why I would venture to guess 99% of all supercritical technology takes advantage of carbon dioxide. So a little bit of history around this as a mobile phase. So in the late 1960s was really the dawn of commercially available chromatography units. And why this is a big advantage is chromatography was around well before the late 1960s. However, once it became commercially available, that's when researchers and academia in several different locations would all be able to then take advantage of that. So the three different types of chromatography or mobile phases that they were concentrating on were gas mobile phases, supercritical state mobile phases, and liquid mobile phases. And I just want to take a pause at this moment to see if the slides are still going. Yeah, okay, we just had a little technical glitch on our end, but I think everybody can still see.
Yes,
so at
we
any
can
rate,
still see it. Thank you.
Thank you for confirming. So each system was characterized again by the mobile phase being used. So it's not really a coincidence that these all came to fruition right around the same time. So in 1967, we saw the first commercially available integrated gas chromatography system. So what that means is it actually had an injector port and a detector all hooked in unison. And for those of us that remember those old systems, we were doing hand injections on big long metal or glass pack columns. But again, right around 1967, you could buy an instrument, set it up in your laboratory, and you were off to the research races. Right around that same exact time, supercritical fluid instrumentation was being introduced, but there were delays. And these delays, unfortunately, when you're talking about commercial scale, can really delay the adoption of the technology. And these delays were really around some calculation errors or estimates around the polarity of the carbon dioxide. It really started to produce results that didn't quite make sense. People couldn't figure it out. Once they figured out they had a factor error around the solvability or the solubility of the carbon dioxide, then they had some first generation engineering issues. HPLC, soon right on the heels of the GC, became commercially available, and then these two really became the first two platforms that methodology started to rapidly be developed on, and then supercritical fluid was playing catch up since then. Incidentally, the science tricorder that those of you who are Star Trek fans debuted in 1967 as well, I don't think it was a coincidence, and I think we all know that if it's on Star Trek, someday it will be true. And I'm just saying right now, since this is being recorded in history, this is what the new chromatography systems will look like probably in a few years. Back to our original slide. What do all three of these images have in common? It's James Lovelock, an English scientist who actually built the first homemade gas chromatograph that took a trip on the Viking 2 mission with NASA back around in 1974, 75 was when the actual gas chromatograph made its trip. But he had created a version of this, and for those of you that are familiar with James Locke, he's also the gentleman who came up with the Gaia hypothesis, where the Earth is actually a self-regulating and self-sustaining system left to its own device and not interrupted by humans on a large scale. The Earth will actually take care of itself and replenish and renew its resources. And we should be working very hard to maintain that balance. Incidentally, he also happens to be the guy who came up with using CO2 as a carrier gas or a supercritical fluid for a chromatographic application right around the same time he was developing gas chromatography, all the way back in 1958. So he really is kind of the father of the supercritical fluid movement, as well as being the first really green dude out there and talking about science does not necessarily have to lend itself to pollution, even though it's required that we use some of these strong solvents and some of these things to study and to research. We can do it in a very responsible way. And he was talking about this all the way back in the 50s, 60s, and 70s. Actually, still is. I think he just celebrated his 101st birthday. If you Google him, there's some really fun documentaries to watch. They're just at least they made me smile. They're really joy-filled experiences to watch those. You can go ahead. Thank you. So why is it a better technology? Well, in future slides, you're going to see that it will deliver the same exact results. However, it's utilizing green chemistry. And there's actually a green chemistry group, if you're not familiar with it, whose total mission is to have international gatherings to promote the environmental sustainability of research and development of everyday chemistry using supercritical fluid extraction and supercritical fluid chromatography. Now, I will say they've made tremendous headway on the extraction front, as this is one of the most popular forms of extraction due to its easy scale, cost, and environmental impact, especially in the botanical and pharmaceutical industries. Now, the chromatography portion is lagging a little bit, but that's what we're here to do today, is to help explain why this should actually be first place in everybody's mind when it comes to chromatography. Takes advantage of the CO2, like we mentioned before. So I've talked about a lot of those big bullet points, but man, let's look at those last two right there. It saves up to 95 to 98% of the solvents used during chromatography, during extraction, and during the projects while you're actively running samples. It reduces exposure to those solvents, not only for the chemists, but for the people creating the solvents, to the people who live around the plants that create the solvents. And what kind of stuff are we talking about? The dichloromethanes, the chloroforms, you know, the everyday reagents and solvents that we're all using, hexane, DMSO. And for some of those complicated extraction processes where we need to use saponification, concentrated KOH. The EPA actually also has guidelines for green chemistry. I know it's a bit of an eye chart, but you can look this up. It's easily downloadable at epa-gov/green chemistry. That's the first government website that's been so easy to access. So we owe them a thanks on that one. And you can see how supercritical fluid chromatography meets several of those bullet points. Why is it better? When the sample preparation, in the particular system we're using, we use an inline supercritical fluid extraction system that's inline right with the supercritical fluid chromatographic system. So we take standard methodology, and I don't think this is an exaggeration to say that there's at a minimum 10 steps in most of them, 15 steps in the average one. This pictogram or this picture is from a Shimatsu brochure on the Nexera unified chromatography system. But you see that there's a significant reduction in analyte degradation elements due to the minimized exposure that you're placing on these things. So there's less time exposing to air and to light, to reagents that are needed to use to create the next step of the reaction, heat that's needed in the extraction phases, something to to liberate things, and all of the transfer steps that introduce scientific error. Now, we all know as trained and validated scientists that it's our job to minimize each one of those touch points. And I'm not saying that we don't do an excellent job at protecting our samples as they go through. But anybody who's developed a method knows the primary goal of method development is to make sure that we use the most simple and least destructive methodology to extract the sample, get it onto the system, and pass it through the detector as possible. That's how we get the highest quality, most accurate results. So this is the actual picture of the entire sample preparation process. It's kind of ridiculously small, which is the whole point, right? So in the time that it takes me to explain this slide, you could actually prepare a sample for analysis. So what we do is we collect that output of a finely ground and prepared sample. And I'd like to just pause here for a second because this is something to highlight too. Sample grinding technology has really made some giant leaps forward into some wonderful benchtop models out there that really pulverize and take samples down to a super fine particle size, which makes homogeneity that much more easy to obtain. And it also creates a little bit more surface area for the extraction to access. So kudos to that industry for making leaps forward, and we can take advantage of it here. Next step is to add a little bit of water. We're using mass spec, so we're using a stable isotope internal standard, and then a little bit of alcohol. You create that slurry. You add a little bit of water absorbing material. So this functions as, obviously the name applies, to reduce the moisture in the sample, but it also acts as a mechanism to increase the surface area for the supercritical, or for the carbon dioxide to access during the extraction cell. Again, it's transferred to the cell, loaded onto the instrument, and that's exactly what the instrument looks like. So brief outlay of how the instrument is set up, and there are multiple, and this actually is the key. Right here is the quantum leap that they were trying to achieve in the 70s, the 80s, and the 90s. So again, applause goes to the instrument manufacturers that have been really working to bring this forward. This is the general outlay of a supercritical fluid chromatographic system. We happen to use Shimatsu Nexera technology for this. The key here is the back pressure regulators. If you remember from the earlier slides, the problem with the robustness of the data and some of the other issues was controlling that pressure. Every time you change that pressure and you move away from that critical point to that sweet spot, anywhere in the system, you're starting to change peak shade. You're starting to introduce background noise, all kinds of parameters that you don't want in there when you're trying to elucidate a peak, right? So that's why people kept going, well, until you fix it, we'll use UPLC. We're going to use GC. We'll just change the detectors, and we'll come up with all these new detector technologies to do what the instrument was supposed to do. But we'll come up with different column technologies. Finally, the pump system, the back pressure regulator, has given us a super tight closed system where we can control the flow of the gas to the fluid through the entire system, all the way through there. And so actually it allows us to divert a great portion of the sample and introduce a totally clean sample into the APCI source of the mass spec. And in this case, we use the ABSciX 6500s for that. And again, the key here is letting the column technology do its job. The column's not supposed to be a filter, it's supposed to be separation technology. The mass spec detector is not supposed to try and sort out garbage, it is supposed to detect your fine peaks. So we're putting on super clean sample, letting the existing technology really get basically supercharged and do the original job it was designed to do. So the first wave of matrices and analytes that we're looking at, we call this the first wave because we think as people start to adopt this technology, we have lots of projects, lots of other matrices to bring on board, and lots of other analytes. Again, on the left-hand side, you'll see there the infant formula powder and the supplement tablets. Those are both certified reference materials that we looked at, and we looked at other supplement formulations such as liquid powders and soft gels. Those really represent the bulk of the most popular things that we see in the fortified food and supplement area. The main analytes that we're looking at and launching immediately are vitamin A, vitamin E, D, and K, and their isomers. And now we're going to go over a couple case studies. So these posters, this slide and the next slide are posters that we've presented at different conferences or trade shows. And it really highlights how we can see current technology, so HPLC, fluorescence detection in the far left there, and the supercritical SFC on the right side. We use two different columns, as you see for vitamin K. The isomers matter. Cis MK7 is nowhere near as bioavailable as trans MK7. So like you who spend a lot of money on your ingredients, you make a lot of investments in your products because you're trying to make it a healthier, happier tomorrow. We want to make sure that we're getting the best form of that vitamin K that you paid for, measured, and to say, yep, you purchased a good product, you created a good product, your ingredients are good, your vendor is now qualified. So this lets us do that. You can see the results are, the results compare almost identically across the board. RFCs, identical. We did some more reproducibility data over there on the right-hand side where we're measuring cis and trans together off a single injection, and then running the columns in unison, I will say, not in tandem. So we're running two separate columns on there to see just the trans peak. And the time it takes us to do a injection on the HPLC, we can actually elucidate those peaks with the supercritical fluid instrument within, I think, 6 minutes. Swapping out columns, we can separate that. And in this slide, you'll see we're looking at two different parameters. How does the method hold up to a certified range on two different matrices? And then how does the method compare to official method settings? So over on the left-hand side, some of you might recognize these two NIST materials. 1869 is actually the infant formula powder NIST, and 3280 is actually the tablet, the multivitamin tablet NIST. And so we measured those two. You can see number of results, average results, and they nested very well right in there in the NIST range, with the exception of vitamin E. I like to focus on that exception. We'll go over to the next column here. Oops, sorry. So we're looking at all the different matrices, again, comparing the SFC to official methodology. And then we calculated a comparison. And you can see they range everywhere from K1 at 93.7% comparability, all the way up to 113% comparability for vitamin E and infant formula. Again, same result, both sides. Why do I highlight that? I speculate, and we haven't been able to push and study on this a little bit, that due to the greatly reduced touch time and analyte degradation that one sees in a standard extraction versus the SFE extraction, or the supercritical fluid extraction, with the sample only being basically exposed from the container to the instrument for less than, I don't know, probably less than 5 minutes, and then it goes through the system in 12 minutes. So you're looking at around 17 to 18 minutes between sampling and measurement versus standard or official vitamin E extraction that is compared to, in hours, maybe 20 hours before it's measured, right? And so we actually pressed on them a little bit. We tried adding the internal standard at different parts of the extraction to see if that varied, and we did confirm that result. So we're speculating that that's actually the real result that current methodology is not able to even, I guess, measure and accurately compute.
Thank you, Richard, for talking us through some of the history around supercritical fluid extraction and supercritical fluid chromatography, and walking us through a couple of case studies. Now, I want to talk about how we validated these methods and why. Why should we switch from official AOAC methods to this new technology? And when we did the method validations for all of the vitamins on SFE, SFC, we met all of the SMPR requirements for these methods as they were validated. We have a lot of methods here in the Madison facility that are AOAC first action, final action, and we really respect what is done and put into those method validations. So we followed a similar suit when we validated these methods. And really, one of the main reasons that we did not or have not yet submitted to AOAC is because this technology is not widely spread enough to really help with a multi-lab validation. And so this is something that potentially in the near future we will be doing. Also, this has been submitted for accreditation for ISO 17025, and we should be receiving that within the month. So how do we go forward, and how do we start analyzing your products using this technology? Well, let's do a mini validation. Let's send some products, let's have them tested by both our current AOAC saponification method and also this SFE, SFC, and show that they're comparable. Results are comparable. And then we can start utilizing SFE, SFC to analyze products in the future. How do we do it? So right now, codes are available. This test is available to order. Go on Sample Connect if you're ordering through the Madison facility, or if you want to send through a different Eurofins lab, no problem. There's codes available on EOL. Just talk to your CSA representative, and you can get these codes added to your quote, and you can go ahead and order right now. Also, one of the advantages, as Richard talked through, right, it is quicker to analyze. And so all of these tests are available for standard turnaround time of 5 days. So same cost as current methodology, but quicker turnaround time to help meet all the needs that are really we're up against in this industry. So thank you very much for listening, and we really hope that together we can create a greener tomorrow. Together we can move forward, use this technology as more of a throughput technology, and we can use this for vitamin analysis. And hopefully in the future, as we continue to advance, we bring on more methods, we bring on contaminants, we bring on amino acids, and we really ask for help in doing that, right? Send us your samples, have a conversation with us. Feel free to reach out to anyone in the company. We can get you started and analyze samples for you as soon as possible.
All right, Thank you so much. I just want to go ahead and launch a poll here just before we move into the Q&A portion. Again, if you would like someone from Eurofins to follow up with you, please select yes. I will go ahead and just leave this poll up for a moment. And again, I want to remind you that you can submit your questions in the question field. There have been a couple that have already come in, so I'll go ahead and just kick us off here with these. The first one is, in order to order, do I have to call the Madison facility?
Oh, that's an excellent question. No, as Marisa stated there towards the end, so maybe that question came in a little earlier than that. You can contact your current representative. How the Eurofins network works is we take care of all those logistics. So you can call the person you're currently working with. They know how to get in contact with the codes. So they have both the Sample Connect and the EOL codes. So nothing really needs to change other than you asking to join the green team.
Great. Thank you, Richard. The next question is, I'm not sure I can explain this to my QA team to explain it to change control team. Can you help me with this?
Yeah, I can take that one, Marisa. Absolutely. So we understand, and that was kind of the point of what I was going in, trying to link all these technologies together and calling this a unified chromatography system. Because the leap is not that large between current chromatography and future. So yes, we're happy to have that conversation. We're happy to tell to speak to your folks. And actually, that reminds me, Amy, I think that we have gotten Dan Hanks, so the developing scientist is going to be available. Is that accurate?
Yes, that's correct, Richard. Thank you for bringing that up. On Thursday, November 18th at 11 a.m. Central Time, Dan will be available. So I do see actually quite a few questions that are coming in that perhaps we will be able to have him answer for us. Again, there will be more details in a follow-up email and communication, but thank you for mentioning that, Richard. Looking at the questions here, and next one that has come in is the question here is, are you willing to work with us on a mini validation?
Yeah, so I'll take that one. Thanks, Amy. As I mentioned before, we are more than happy to do side-by-side comparison at the cost of Eurofins to show that this technology does indeed give the same result as current technology, maybe even a little bit better precision. So please get in contact with anyone from Eurofins that you're used to working with, and we can make sure that we are in touch with them and we get the right validation for the type of work that you need, for the samples that you need, and we can work together to accomplish that. So yes, more than happy to work on what is needed to bring quality, bring your team along for a mini validation.
Wonderful. Thanks, Marisa. The next one again here is, are you controlling the full system with one software?
Yeah. Yes. Yep. That's accurate. I believe that is the case. And so Dan will actually go on Dan's talk. He's going to actually go into a little bit more detail as far as instrument control, any of the challenges that we see around data acquisition and things like that. But yes, to my understanding, that is accurate.
Thank you for that, Richard. And I think we have one more question in here before we wrap it up. Someone has a question. How far along are you on the pesticide testing by way of supercritical fluid extraction?
So that's a great question. So we have done a little bit of preliminary work in looking at some of the pesticide screens. I would say that we are at this point we are kind of gauging interest, and really need to show some success in other applications before we bring more online. So if you are interested in applications for pesticide analysis, please, let's have the conversation, and potentially we can do some side-by-side comparisons with our method development chemist, and we can move that up in the priority for working on next application.
Right. And
Wonderful.
really, we are super happy to do that. But again, we need folks to come along with us on this journey. So it's not just about, you know, send your samples to Eurofins. It's about adopting this technology. And so we really do need partners to help us to bring this, what we believe to be a better technology, safer, greener, you name it, to the front. We need partnership in that.
Wonderful. Thank you so much. And again, I just want to remind everyone, again, this is pretty much wrapping up our Q&A session. Again, if there are other questions that do come in, we certainly will address them offline with you. We invite you to attend that webinar in a couple weeks here with Ask the Scientist, Dan. And again, I want to remind you that you will receive a copy of the recording along with these slides within 3 business days. So thank you again, Richard and Marisa, for your expertise and presentation today. And thanks everyone for attending.